The function is also the ultimate trait that dictates the selection of organisms. If an organism cannot function in a certain environment, it cannot survive. My name is Marian Hu. I'm a marine biologist and physiologist. The trend in life science is partially moving away from this field of traditional physiology. Now in the times of omics, where genes are screened and proteins are characterized in 10,000s, it seems a little bit, inefficient, this work that I do. But I think on the long run it pays off, because to understand how environment or environmental changes impact organisms, we need to understand their function. In particular, I'm interested in how animals maintain environments within their bodies, but also within their cells. And here, membrane transport physiology has very important implications, for example, for the process of calcification. I'm working with the sea urchin larva as model system, which has been used by developmental biologists since over a century. Ocean acidification research has focused on the calcification process in these animals. The idea was that this acidification must impact these calcifying skeletons. And this is why a strong focus has been dedicated to understand physiological processes in the sea urchin larva. There are huge challenges in my work, namely to develop new methods to study very sophisticated small processes inside of the cells of very small organisms. And I must say I'm very fortunate to be associated with the Institute of Physiology, so with the medical faculty, so I could adopt methods, state-of-the-art methods from the medical field and use them to study my research questions in marine animals. So on the organismic or the cellular level, we mainly use live cell imaging technologies to monitor pH or ion concentrations inside of cells or even in subcellular compartments like vesicles, to understand how these substances or ions are transported across membranes of these compartments. Another very strong method that we use is electrophysiology. Here mainly ion-selective electrodes. So these are very small electrodes with a tip size of about 1 micrometer that we can insert into very small extracellular compartments, but also intracellular compartments to measure ion concentrations, pH, and membrane potentials, for example.